In our fifth podcast on the renal system, we're going to talk about the renal tubules and the collecting ducts. Now, as you appreciate, the filtrate or urine gets modified as it flows through the nephron and the collecting ducts because of the different properties of the cells along the pathway. Now, we're going to look in turn at cells of the proximal convoluted tubule, cells in the proximal tubule through the loop of Henle, cells in the distal tubule, and then we're going to look at the principal and intercalated cells in the collecting duct. So this diagram is just showing what you already appreciate, the path that the filtrate travels as it's made in the glomerulus and collected initially in the urinary space of Bowman's capsule, and it's going to go through the various tubules like so. And here we're all still in the nephron. Then when we get to these regions of the distal convoluted tubule, the initial collecting tubule, this is where all the tissues that I've traced this way would be one embryologic origin, and then here a different embryologic origin for the collecting duct system. So this is just the pathway that the urine travels ultimately to the ducts of Bellini. Let's talk initially about cells in the proximal convoluted tubule. This is the initial and major site of reabsorption. It is also a site of significant secretion for non-filtered substances. The first thing I want you to realize is that material can go either way. From these proximal convoluted tubule cells, can go from the tubule to the interstitium to the blood, or can go the other way around, from the blood to the interstitium back into the tubule. At the proximal convoluted tubule, about 80% of the sodium chloride and water is reabsorbed by these cells. So in the initial filtrate, 80% of the sodium chloride and water are reabsorbed. In the filtrate, essentially 100% of glucose, amino acids, and proteins are absorbed by cells in the proximal convoluted tubule. These are simple cuboidal cells with an extensive microvillous brush border, basolateral infoldings in these cells with many mitochondria. They have an active endosomal and lysosomal compartment, and you might remember that acid phosphatase is a marker enzyme for lysosomes, and so you might expect, if you were doing cytochemical stains for different cells in the kidney tubules, the proximal convoluted tubule cells would stain more intently for acid phosphatase. These cells have very extensive fluid phase pinocytosis. There's a sodium-potassium ATPase that establishes a gradient for secondary active transport, and these cells exhibit extensive tight junctions in their apical regions. And this is an electron micrograph of the proximal convoluted tubule cells. Look at the very extensive microvillous brush border. Look at the numerous mitochondria and structures like lysosomes. At the light microscopic level, the cells stain very intensely with PAS stain because the glycocalyx of the brush border contains various peptidases and disaccharidases. Here you can see the brush border staining positively. These peptidases and disaccharinases are going to break down proteins and sugars that get into the filtrate, and that will make it easier for reabsorption to occur. Now, I would like to stress that these cells are going to reabsorb material that's in the lumen. They're going to reabsorb this way into the cell, and then we'll get back into the blood. But you might also appreciate that substances that can't be filtered might be excreted by the proximal convoluted tubule cells in this direction. So excretion could be in that direction. Reabsorption could be in this direction. If we consider cells in the proximal tubule through the loop of Henle, this region of the kidney tubules, these cells are less and less specialized for reabsorption the further you go from the urinary pole. So you can talk about cells in the straight segment of the proximal tubule through the thick descending limb of the loop of Henle. As I said, these cells are less specialized for absorption. Then you move from the thick descending limb to the thin descending limb. These cells are permeable to water. And remember, the length of the thin limb will vary with the location of the nephron, whether the nephrons originate very high up into the cortex as short loop nephrons, or whether they're juxtamedullary nephrons, the so-called longer loop nephrons. And remember, these longer loop nephrons play a greater role in conservation of water. But the important thing I want you to remember at this point, the thick descending limb of Henle's loop through the thin descending limb, these are cells, the cells here are permeable to water. If we look at cells in the distal tubule, we could be considering cells all the way from the thin ascending limb of Henle's loop to the thick ascending limb of Henle's loop through to the distal convoluted tubule. If we consider these cells, the thin and thick ascending limb of the distal tubule, the cells are impermeable to water. These cells can actively reabsorb sodium under the influence of aldosterone. Chloride will follow the sodium. These cells can also secrete hydrogen ions and potassium. This is just a histological view showing some of the distal tubule cells versus the proximal tubule cells, which are more eosinophilic. So the distal tubule cells, they're less eosinophilic, they're smaller. As a result, you can note more nuclei in cross-sectional profile. And in the kidney cortex, the proximal convoluted tubule cells tend to outnumber profiles of the distal tubule, and that's because the proximal convoluted tubule is about 6 to 7 times longer than the distal convoluted tubule. Now we can look at cells in the collecting ducts. These collecting ducts begin in the cortex from the distal convoluted tubule, and collecting ducts then descend into the medulla as the papillary ducts and eventually the ducts of Bellini. So here, as you follow from the diagram, you've got from the distal convoluted tubule, you've got cells of the collecting tubule, then the initial collecting ducts, the cortical collecting ducts, the outer medullary collecting ducts, and then the inner medullary collecting ducts. And the diagram shows how the morphology of these cells changes a little bit. I'm not going to ask you to worry too much about the differences in morphology of the cells along the collecting tubules and collecting ducts. But what we can say, the cells in the collecting duct tend to be simple cuboidal to columnar cells. These cells are always impermeable to water unless ADH, antidiuretic hormone, or vasopressin, is present. There are two major cell types that we talk about in histology in the collecting ducts. There's principal cells, also called light cells. These cells secrete potassium and they reabsorb sodium under the influence of aldosterone. And there are the intercalated cells, the so-called dark cells. These cells reabsorb potassium and they regulate acid-base balance, and they play a role in transport of hydrogen ions. This section is deep in the medulla, and the cells here are primarily principal cells. Here on a histological view, we're deep in the medulla, so we're showing primarily principal cells or light cells. On the longitudinal view, you can see actually some of the intercalated cells that actually are staining a little bit darker. And here you can see the cells. Again, this gets at a little bit of the morphology that I'm not going to ask you to worry too much about. But as you're in the more proximal regions of the collecting ducts, the cells tend to be a little bit flatter. As you go more distally, the cells get taller. Again, I'm not going to ask you to worry too much about that. This is a scanning electron micrograph showing the intercalated cells, or the dark cells, that have these extensive microplicae or ridges on them. Here they're shown in a little bit higher power. We don't know what the function of this extensive microplicae. It may have something to do with regulating acid base, but we're not really sure about that. And then the principal cells, or the lighter cells, have very short microvilli on them, but they do have a single solitary cilium, which you can see standing out like this and like this. We don't really know what the function of that single solitary cilium would be. Here's a region in the outer medulla of the kidney. You can see thick and thin limbs of Henle's loop. You can see the vasa recta, and you can actually see collecting ducts that show light staining, or principal cells, and dark staining, or intercalated cells. This is a nice example right here of some of the darker staining and lighter staining cells. Now, just a little bit about the histophysiology of the kidney. Here we can remind you of the fact that all the way through the thin limb of Henle's loop, through the distal tubule, through the collecting duct here, these cells are impermeable to water. So what happens as water is drawn out in the early parts of the nephron, but then sodium chloride drawn out in the later parts of the nephron and in the collecting duct, the water remains because these cells are impermeable, so you make a very dilute urine. On the other hand, in the presence of antidiuretic hormone or vasopressin, aquaporins can be inserted into the membranes of principal cells, the so-called light cells, and under the influence of antidiuretic hormone, water can be withdrawn from the filtrate as it passes through the collecting duct, and as water is continually withdrawn, you get a more concentrated urine. Now again, physiology is going to go into this in much greater detail, so here I'm giving you just a general overview to help you relate the physiology to what you'll see in the laboratory here for histology. The vasa recta plays an important role in maintaining the osmotic gradient in the medulla by countercurrent exchange between the atrial and venous sides of the vasa recta. The atrial side of the vasa recta has the ability to lose water and gain salts, whereas in the venous side of the vasa recta you tend to lose salts and gain water. This is a very unusual type of arrangement, but this countercurrent exchange tends to help maintain the very high osmotic gradients as you go deeper and deeper into the medulla of the kidney. And again, this will be highlighted in much more detail in your physiology course. Here is just a very, very low power view of the kidney, and I just wanted to point out what a renal pyramid would look like. So I'm looking at a renal pyramid like this as I would trace the renal pyramid, and you can see profiles of some of these collecting ducts which would be draining into the calyceal spaces down here. And here, just at a little bit higher power view, you can see these ducts of Bellini that would be emptying into the calyces of the kidney and eventually into the ureter.